This is a strand of DNA, and this is a picture of Johann Friedrich Miescher. But what is the similarity between the two? The answer is, you can edit both of them. Yes, you can edit DNA like you can edit a picture. This concept is called gene editing, or also known as genetic engineering. Gene editing involves the manipulation of genome makeup using biotechnology. Nowadays, modern gene editing primarily uses CRISPR as a method for this practice. Now let's go through the brief history of gene editing. Back in 1960s, scientists began to find ways to edit genomes. Recombinant DNA was introduced in 1972. Then, in 1989, gene therapy was introduced. More than a decade after, zinc finger nuclease was created to facilitate targeted gene editing in the 2000s. Finally, in 2012, CRISPR technology was invented to increase the productivity of gene editing. But how does gene editing works? So the first step, get a lab. Don't do this at home. Second step, give a copy of DNA that is modified to the CRISPR system. And lastly, put the system into a living cell. Now, we have introduced gene editing, its brief history, and how it works. Let's now get into the heavy stuff, starting with its relationship to religion. The relationship between religion and gene editing could be quite complicated, but in summary, most of the religious groups view this practice as a violation against their belief, because it seems that we're trying to artificially create a new form of life, which shows that we want to imitate how God creates life, in the sense that we're not satisfied with God's creation. However, the relationship between gene editing and humans could be traced back to the prehistoric age. When our ancestors began to practice agriculture during the Neolithic era, the agricultural crops which originated from wild plants also started to display a different traits from the originals. In the long term, the genes carried by the domestic plants would be very different from the wild plants. Furthermore, when our agricultural technology becomes more developed, we started to use cross-pollination and cross-breeding to control the genes inherited by the crops and livestock. But essentially, we're selecting the desired gene and only allow the specific gene to produce, and that's how the fundamental of modern gene editing technology works. Now, let's get into the moral and ethical views on gene editing. Technology is useful if used in the right way. Gene editing is no doubt a major step of advancement in the medical field right now. However, there are a lot of controversies on this particular topic. Gene editing is a two-way sword that can let us speed up the advancement of science and technology, but it can also act as a powerful weapon if used in the wrong way. Bioterrorism is such where terrorists may use it as a weapon to assassinate someone or to control the world. There is no clear line in gene editing being legal or illegal yet, and people may take advantage of these blurred boundaries. Besides that, experiments on animals are also a concern for animal cruelty, whereas experiments on plants will bring up the concerns of pollution and mass genocide of species. Gene-edited babies to create the perfect babies also raise up moral and ethical issues in the community. Last but not least, doubts about the cost of gene editing, where it is also for the rich or for everyone. We humans have to think about the ugly side of gene editing and find solution before we really implement it for the greater good of mankind.
Hello, now I'm going to talk about the role of gene editing in life. Gene editing can help animals by improve their quality and to avoid them from carrying any disease. Scientists have produced gene-edited animals they say could serve as superdad or surrogate sire. The researchers used a high-tech gene editing tool to knock out a male fertility gene in animal embryos. The animals were born sterile but began producing sperm after an injection of sperm-producing cells from donor animals. The technique would enable surrogate males to sire offspring carrying the genetic material of valuable elite animals such as prize bulls. For example, bull superdads could be used to sire healthier or mature cows. Lastly, gene editing can help plants to become more notorious and will able to grow in extreme weather. Gene editing in plants is usually achieved by modifying the genome of cultured cells and then regenerating whole plants by exposing the modified culture cells to growth hormones. This method is inefficient, limited to a few species, and can also lead to undesired genome aberration.
Thank you, Din, for talking about the roles of gene editing in life. So, in conclusion, genetic engineering still has a long way to go, akin to the internet in the 90s. Society shall decide whether the practice of genetic engineering should be given a green light or not by weighing the pros and cons of this practice. Thank you for watching.